Good afternoon, everyone. It's nice to be back with you to do a bit of life science with you today. I trust that you are well, and I hope that you are already studying and prepping for the exams. The section of work that we're going to have a look at today involves blood grouping and paternity testing, which will probably be the basis for the first session that we do. And then the second session that we do will involve DNA fingerprinting and genetic engineering. So what I want to do just first off is just recap a little bit of work that links to the genetics section that you have to know before we start with the actual blood grouping and the paternity testing, because there are a few terms that we have to know. If you don't know them, then it makes the blood grouping very difficult. Right, so the first terms that we need to know are those of homozygous. Okay, a homozygous condition is one in which you have two of the same genes controlling a characteristic. Okay, so you're either going to have two recessive genes or you are going to have two dominant genes. Then heterozygous is the next term. Okay, heterozygous means that you're actually going to have one of each expression of a gene. Or you can say that you're going to have a different expression of the gene. Okay, so if we have to write this down, there you would have one dominant and you would have one recessive gene if we use the letters B to start with. Then the other terms that we need to know, which I spoke about and I mentioned, is that of dominance. Okay, dominance means you have a gene that when expressed will be seen in the phenotype of an organism. Okay. Another thing that you can say about whatever gene is dominant is that it often overshadows the recessive gene. Okay, so we are going to see this in the organism's phenotype. Okay, and just again, if we link it back to the heterozygous and we think of dominance, okay, dominance can be expressed either as a homozygous condition, all right, when you have two of the dominant genes together, but if we are speaking about the individual's phenotype only, that is also going to result in the same phenotype as having a homozygous condition. Right, whereas if we speak about recessive genes or recessiveness, okay, that is a condition that can only be seen if both recessive genes are expressed at the same time. Okay, so to have a recessive condition, the only way to have a recessive condition is to have both of the recessive genes together. The minute you have one dominant gene and one recessive gene, it's going to change the phenotype. Okay, so the recessive gene is something that is overshadowed by the dominant gene. And then the last two terms that are really important for this section of work is those of phenotype and genotype. Right, so an organism's genotype is its genetic makeup, or you can say its genetic composition. Okay, and can you see all of these terms are kind of synonymous with each other? They kind of interlink and they really have to be used very carefully and very closely together. So if we again have to use letters, a person's genotype is the thing that you express by looking at what genes they have. So they can either be homozygous and dominant, or they could be homozygous and show the recessive condition, or they can have a split gene composition, okay, where they have one of each. They've got the dominant and the recessive gene, and then they have the heterozygous condition. So the genotype denotes to what genes does this individual or organism have, whereas the phenotype is going to be the physical characteristic that we see in an organism. Okay, and here you're going to have answers that would be something like brown, or really tall, or short, or pink, or anything like that. So the phenotype, even though it's the physical characteristic, and there's a nice way to try and remember it. Okay, the pH of phenotype shows us the physical characteristic, whereas the genotype, okay, that and that is the same, is going to tell us about the genetic makeup of an organism. Now, the thing to know here as well is that the genotype of an organism is going to determine its phenotype. Okay, so whatever we have as a combination here, it is going to definitely influence and determine the phenotype. So the thing to look for again, all 6 of these terms. Okay, if we look at homozygous and we look at heterozygous, we look at dominance and recessiveness, and we link it to the genotype and the phenotype. The genotype is going to be expressed with capital and lowercase letters in the case of complete dominance. If it's incomplete dominance, okay, and codominance, which we'll get to in a moment, we have to actually use 2 different letters, and they are expressed by using them in capital form. But the genotype of an organism is going to influence and determine its phenotype. Okay, this genotype and that genotype are going to give us the same phenotype. So the genotype is different. Okay, we can see that that is a homozygous condition and that is a heterozygous condition, but they are actually going to give the same phenotype, whereas the way in which we are going to see the recessive condition in an organism, the only way we're going to see the recessive condition in an organism is to have both recessive genes together. Right, so those are the first few terms that we have to know. With that being said, now we can have a look at blood typing.